My name is Corentin Cras-Méneur and I'm a research assistant professor in the Division of Metabolism, Endocrinology and Diabetes at the University of Michigan. I work in the Brem Center for Diabetes Research here in Ann Arbor, Michigan. My original training was in molecular genetics and developmental biology, and I've been working on diabetes research for the past 16 years. I lost one of my grandfathers to diabetes when I was only 8 and decided very early on to devote my career to diabetes research. A lot of my work focuses on developmental biology because I truly believe that understanding how beta cells are formed during development could be key to generating new beta cells for diabetic patients. I also have a lot of interest in beta cell loss and regeneration. As you can see on this diagram, during the early onset of diabetes, a number of cells producing insulin are progressively lost. The few remaining healthy cells that have not yet been destroyed are then put under tremendous stress to try and compensate for that loss. A number of investigators have devoted their work in an attempt to stop the progression of the disease, and a few recent studies have now indicated that the progression of diabetes could be aggravated by the fact that these few healthy cells that have not yet been destroyed are put under such a heavy load that many of them stop producing insulin altogether and lose all the characteristics that make them insulin cells. As you can see on this picture from one of our genetic models, when you put the islet cells under stress, while some of the cells can still express fairly high levels of insulin, here in green, many of the other cells seem to progressively fade away and stop producing insulin. This process is called dedifferentiation and could affect not only the healthy cells in diabetic patients, but also the cells from grafts from donors or the chances of success for cell therapy. Working with some of our collaborators in the Brem Center for Diabetes Research, we have had the opportunity to work on different models of beta cell dedifferentiation, and we have now identified steps that could stop this process. The experiments are currently well underway, but the careful analysis and manipulation of these pathways is unfortunately costly, and this is why we need your help. Please donate through the Diabetes Research Connection and share with your friends and families.